Fantastic, thanks. I should probably also mention that the theme for our panel discussion is transforming care from diagnostics to prevention through network collaboration. And I would like this to be conversational, I think. So, and again, very quick pace this morning, so I think we can slow things down and have a conversation about transforming care. Would welcome questions from the audience. I guess I'll exercise the chair's prerogative while people think of their questions to ask one myself. And I'll sort of open it up to any of the panelists. Do you see particular research or policy or programmatic priorities that have sort of immediate near-term translational impact that you think are particularly fruitful for the consortium to emphasize?
Before we started talking to Adam, and we had talked about that before, I would say two things. One thing that I have been saying for a while is really the use of the clinical data. We have millions of patients that have been sequenced in US and other countries, and this data is sitting in clinical labs and very little use of it. The sequencing is done today, the report is out, this data is never used again unless we as physicians request a reanalysis that is done once, and then that's it. Some hospitals like Hopkins are requesting the files to be stored in a database at Hopkins and be reused later for clinical and research use, but that's very few hospitals are doing that, all in silo. So I think come up with a smart way to get that data to be used more in the research in the clinical side. That is, I think, one of the things. The other thing is on the cancer field, and I'm not a cancer field researcher, I'm a rare disease researcher, and that's why I started with the germline sequencing. But usually when we talk about cancer, we think about somatic sequencing and not so much the germline. But I think that's another opportunity for us to investigate further and try to find susceptibility genes and variants. So I think we have to start thinking of that more. In my case, we started actually with the germline, trying to find the susceptibility genes and variants. And then we are putting that together with the variants that we find in the somatic sequencing of the tumors of the patients. And I think that's another way to explore the research in cancer. I think these two things come up to my mind as things that we can explore. And also, of course, data sharing. That I'm going to stop and give opportunity to other people to talk.
I can actually just jump right off of that because I agree completely with the data sharing, specifically the germline data. I mean, I think we think a lot about the somatic, but we don't always think about integrating that with the germline. And they actually inform each other really nicely. We've really been able to learn more about which germline variants are important based on what they are doing in the tumor and vice versa. And then we say right now that 15 to 20% of patients are predisposed. I think we all think it's much higher than that. And we're not going to identify those predisposition syndromes until we are sharing data enough to find those rare syndromes.
So I agree with Nara on the making the data, especially the clinical data now, available for research use is really important. And Vincent laid out a scheme that Quebec, or he's working on for Quebec, which is even cooler, that really looks valuable and should be pursued. The other thing I have thought about as being very useful is still controlling, who can see the sensitive genomic data, because you can't make privacy, absolute on that, but controlling it in an intelligent way, but still figuring out a way to make citizen scientist types, like motivated parents of patients, able to access that data, because you can unleash a whole lot of talent for solving research problems that way, if you can do that and still keep the confidentiality and assure confidentiality, et cetera. But I think those two movements would be really valuable.
I get to go last. I think I'll just make the point that I think one of the things we've worked to do at CBTN, and I think now we've got more opportunity than we've ever had. I mean, we were told you're going to cure cancer if you generate data. So our field, I mean, this was a dream to CNS, and a lot of us, sequenced everything we could get our hands on. I mean, we were talking about how medullos probably had more medullos sequenced than a lot of way more common tumors. So I think we did that as a community, and I think what we are now doing is how do we actually use all that data, and some of the non-big data, like some key clinical things, are actually where the challenges, at least I'm focusing on, and I think CBTN is ideally positioned to try to demonstrate how to do this. We actually thought, or I thought, that part would be easier. It's a lot of work. It's not traditionally funded or glamorous, and I think trying to work on these new initiatives for how to actually support some of the not AI, low-tech data sharing and making sure we do it right. And I mean right in that it matters, and we have to remember these are patients. So that's kind of where I think we're positioned to walk across the sort of understanding of patients to those who understand much bigger data.
That's a question, Adam, I think. Go ahead.
And it inspired me as you guys were talking, and Sarah was really sort of surprised, and that with whole exome you saw such high, essentially reported incidence. I'm always being asked, where are we in terms of like whole exome versus whole genome? And I think this panel probably could inform the context. I think in cancer we're still panel-based and whole exome maybe. I feel like on the constitutional side, everybody's really pushing hard for whole genome minimally. I think do people think it makes a difference, or what are your thoughts?
I particularly think that in the clinical side, genomes as they are analyzed nowadays don't make a huge difference. In the clinical side, on the research side, we have to be doing genomes because we need to understand so they can inform the clinical side. And we are not going to understand non-coding variants if we don't do that on the research setting. So unfortunately, I think when we get our funding, we still want to sequence as more patients as we can, and we end up doing exome because of the price difference, because we can sequence more people. But unfortunately, I think we have to start thinking of that and sequencing a little less and more of the genomes, because a lot of these susceptibility variants, they're going to be non-coding, and we need to understand them. And to understand them, we have to do sequencing of the genomes. And then there is long genome and the other things that we can explore, structural variants, repeat expansions that are so little explored as of now. So I think hopefully the price will be right and long read can be used more in the research setting sooner than later.
So we've seen the price since I got involved with Kids First. We've seen the price of whole genome sequencing drop to now about what we'd be charged for long read sequencing, and the cost for whole genome sequencing, the Broad quoted the program was $350 a genome. Now, yeah, it's more data that you've got to deal with and you've got to learn how to deal with than just exome, but at least as a biologist, all of the cis-regulatory elements that you would be missing from just exome, it seems like a no-brainer. And $350 is like cheaper than most diagnostic tests. I don't, it's got to move that way, and then hopefully to long read as well, because there's a lot of variants. What the last I saw was at least 25% more variants that are identified as being, possibly indicated.
And then regulation that you can analyze the
regulation. Right. Anyway, it's harder to deal with, but I think, that'll happen. People like Allison will make things happen, make it so that people can analyze things. And, we'll figure it out. So I think it's just got to go that way. And especially because any of these efforts should be toward building a resource of data, and that child's DNA should be contributing to the future research efforts, if possible.
I'm in favor of more data and repeating and doing the same patient on multiple things. I mean, we never sit down at our molecular meeting and look at one patient and don't learn something if we did a new test.
So I'm hearing not only is there a need to generate more data, new types of data that could potentially address, genetic determinants and other factors that contribute to cancer risk, but there's also a need for better curation of that data. And so it's to sort of succinctly and clearly communicate to practicing oncologists and physicians, what tests need to be done, what are the implications of positive or negative results for those tests. Yeah. And that raises questions too, I think, about how we accomplish that. I've heard talks today about, upskilling our workforce, to ready them to face these challenges, but analysis of genomic data, machine learning, these are very complicated disciplines, especially for somebody who's spending a lot of time in the clinic and can't dedicate years of their life to picking up these new skills. So I wonder, how do people see our group and other collaborative groups facilitating this? Is it upskilling our current workforce? Is it really more about building collaborations and bringing in data scientists and computer scientists who can ask these questions? We're talking a lot about sharing data, genomic and otherwise, but do we also see, the Kids First initiative and the CBTN as mechanisms to share workflows and technical expertise through, Dockers or other mechanisms that might make it easier for people to make this leap? How does the panel think we move forward with actually, interpreting all this data that we're going to generate?
Yeah, so I have always been very interested in focusing right in the middle where someone says you can't be, because I think, it's sort of where are those gaps that we all talk about, and we become, expert on one side or the other of this work. And I think what's fun about CBTN is trying to pull this whole spectrum. And sometimes we're a little disjointed because of that, which I think is okay and important. But what I mean by that is I think we are doing both things that are clinically relevant. We're talking about data that is absolutely clinical. Patients were treated based on the expert opinion of their physicians. That's medicine and somewhat scientific, not always. And then, on the other side of like discovery science, where we're talking about technology that you absolutely are not at all ready to apply to the clinic. But what I've seen is this kind of, if, where we were 10 years ago and where we are now, even over the course of CBTN working on this, is we've seen all these things actually translate from where they were researched to where we can figure out what is actually clinical now. And so I think that's where things are most exciting. And things like whole genome and long read, they're already there on the edge, right? There were reasons whole exome was picked for the MCI as the platform that could be already fully clinically returned as a test. But the other data, that's not at the expense of the other data, right? Because the DNA is there, whole genome's happening. I'm super excited about long read, and I know very little about it other than it sounds like magic to me right now. But I want to do that, and I'm already partnering with some clinical labs that are getting that up and running clinically. And so I think exactly where those edges are, when do you draw the line and say, this was research last year, but it's now clinical practice. I think those are most rewarding experiences across the whole spectrum for all of us, no matter what part of that spectrum you're on. So that's why I currently haven't left my CBTN role in terms of wearing too many hats, because I think we're kind of most positioned to have that conversation about being able to say, one foot is totally, this is pure research. I know I can't use it for patient care, but maybe next year.
So I think there needs to be a lot more tool development to enable people whose expertise is not primarily bioinformatics to use the data so that clinicians can use it. And their expertise is going to be treating the patient. That's an art, not necessarily a science, but that's a real expertise. And I don't think you could probably reasonably, or humanoids could really do both, but But we can develop tools, some of which involve AI, some of which don't, but we can develop tools to make the data more useful to everyone, both research on the research side and on the clinic side. And honestly, I think there needs to be a concerted effort to facilitate valid return of results type reports, to using not just the 3 indices that somebody happens to be familiar with, but using all the data that's available and up to date, as well as continuing, I sort of had envisioned a center where people were both doing research on what's the best way to develop these tools and to return results, as well as generating the reports back to the clinic, as well as when new information that's actionable comes in, updating that, the physician, the clinician. That was sort of my pipe dream, and maybe that'll happen.
I was just actually going to jump off of that because I think that the way that we get these more diverse data sets is by making it easier for many different people around the world to analyze their data and enabling the sharing. I think we heard a lot about tools to do that are really, really exciting. And the way that we help people, not just nationally, but internationally, to capture those rare variants and understand them better is by making that data easier to analyze and interpret. I think also, just as somebody who comes from the clinical side, I think the data is coming to clinic. I mean, I think we're doing a lot of sequencing. I think the MCI is a good example. I think there are things that we, in our internal testing, we don't know what to make of, but I think those mechanisms to update our interpretations over time for the families are really, really important. And there's a lot of smart people on all sides working on this, and I think it is about those systems to connect them. So,
yeah, and the only thing I will add is when we talk about making it easier to access, it's not only the big data access that is also important, because as we heard here before, trying to get access to the raw data through dbGaP is not an easy task. It's not impossible, but it's much better than it used to be, but still it's not easy. But if we want to get clinicians, healthcare providers in general, accessing data, we are not talking about raw data. We're talking about different levels of data. So when we're talking about sharing data, we have to think of what kind of data and what level of data to whom. And that's very important to get a variant level. I want to know who has that one variant and what's the phenotype of that individual that has that one variant, or what are the individuals that have variants in this gene. So different levels of access is very important also, not only to think about all the genomic and omic data so someone can analyze it all together. We need that, but we also need to go a little easier on what kind of data we are talking about. On one of the other sides of my research, it's actually working with data sharing, and I don't know if anybody here has heard of GeneMatcher, but GeneMatcher it's 10 years now. This year is the 10th anniversary, and it has 20,000 users in 100 countries. And basically, you go to GeneMatcher and you say, I'm interested in this gene X, because I was investigating a patient with intellectual disability and I found this gene as a candidate. Now I want to find who else in the world is also interested in this gene. And you go in GeneMatcher, and GeneMatcher will put you in contact, and it's simultaneously, so everybody has access to each other information right away. Basically only tells you, I went there, and it tells me that Adam also is interested in this gene, and gives us our contact information to each other, and we can contact each other and go over the phenotype. Sometimes it's not a clinician and a researcher with another patient. Sometimes it's a mouse model researcher. Sometimes it's someone who is doing functional studies and trying to understand that pathway, and it's interested in that gene. So now you put the data together from both sides and decide if that's a good candidate or not. It has been very successful. It has been enabled more than 800 publications that were cited. I don't know if more than that, but more than 600 novel genes have been discovered with the support of GeneMatcher. So we have the new version of GeneMatcher is now variant matcher to the variant level. And these databases are connected to other databases around the world that have the same kind of information. So when you go to one of them, you can actually find if that gene or that variant is also in other database around the world. So we have developed these APIs as part of other projects like Matchmaker Exchange and GA4GH that I don't know if you have heard of, but that is the kind of level that we are talking about and the kind of connections that we are talking about, not only the researcher dealing with the big data, raw data.
Okay, so no other comments from the panel in regards to that question, I take it. Just a reminder, we do have a gentleman in the back with a microphone. If you have your own questions for the panel or would like to add some comments, feel free. And if you would like to ask a question in the chat, I can pose that to the panelists as well. On the note of using all the data, our discussion is sort of heavily focused on generating and operationalizing complex molecular data. It's on the cutting edge, right? But I don't think we have a very complete picture of how structural determinants of health, people's social circumstances might impact cancer risk and prognosis, particularly in pediatrics. And I'm curious to get the panel's thoughts on whether we as a group are doing enough to collect the sort of data that would facilitate that research. No takers, huh? Pass.
Oh, you can start.
Yeah, I can just say that short answer is no. I don't think we're collecting enough of that information, but I do think that there are some pretty significant efforts in play. I think through the COG are the ones that I'm the most familiar with, but also there are efforts in the adult world to, I think specifically of BRCA carriers to better understand how social determinants of health are affecting cancer risk. I think that we could certainly incorporate that more into our clinical information gathering. I think it's just about making it systemically available within those studies, which I think is coming. I don't know if you have more to say about that.
Yeah. No, I think that there is interest in collecting and submitting that kind of data on a broad scale. It's been attempted in multiple ways in COG, and I think it's been the barriers have been that we as a country don't have a way to, it's not clear whose job that is to collect that data, right? So it's not therapeutic trial data, except for it's associated with survival as strongly as almost any other data elements you can pick. So I think when we talk about, it's hard. Any of us who are face to face with patients see all the things that are not medical and not biology that affect their outcomes. And there is good data on that in the context, particularly of leukemia, where there are larger trials and you're able to pair those data elements. I mean, they matter more than if you got the drug in some cases. Some of the kind of data that we can select, and there are validated ways to collect social determinants of health data. Some hospitals have started doing that across the board as a hospital system, not for research, but to know what we need to know about patients in a uniform way, which I think may end up being how we get that data. But I think it's also for us there's an advocacy issue to say, I don't care whose job it is, but we should collect it. We should hopefully use a mechanism like the Children's Oncology Group to reach all these patients that may not be in our academic institutions. But yeah, we need to do it. But the conversations we've had within the Children's Oncology Groups are somewhat infuriating because we try to sneak it in each trial because it doesn't quite go. And there isn't as much. So something like getting it into something super broad like Project Every Child, where it doesn't matter whether you're on a therapeutic study or not, I think is really important too.
Yeah, I've seen some encouraging developments, to me at least, like in the field of ALL, like Kirabona's work, for example. So
a thought related to that is that there really needs to be the development of community-based standards for making that data so it's collected in the same way and expressed in the same way. And really that's something that should be organized for the community, and there should be a handbook to say, if I want to figure out how many aspirin the kid had, this is how these are the questions I ask, and then, just some standard system that the community agrees on. And that's not easy to do, but it has been done for maternal health. at any rate, and recently. And Adam's group was involved in that. And I think another thing that could be done is to do that for pediatric data, not just pediatric cancer, but for pediatric data in general, would be very valuable.
Yeah, it occurs to me, so no tool is going to be perfect, right? Particularly because these are such complex constructs, but the Phoenix toolkit, if you're familiar, might be of use to us here. I haven't logged on in a minute, but this is an NIH resource, basically a compendium of different sort of validated questionnaires and measures that can be administered to study participants to help ensure that we're collecting robust data, and as much as possible that groups in different locations are collecting the same data to try to make it interoperable. So
To me, it's so, I can't understand why we can't do it in an easier way, because every hospital is using electronic medical records, and most of the hospitals are using the same platforms. So who do we have to bring to the conversation to get Epic set up? So Epic is collecting the data in the same way, and this data can be easily used for research. Because, I don't know if it's Epic, if it's the hospitals that we are using Epic, who in the hospitals. I know that we can get the data from Epic to do research, but why can't we share this data across different hospitals or collect from different hospitals and then do it? That should not be that hard at this point. Everybody has it already. Someone has it. Well, is it?
So I'm hearing from the audience that the major sort of barrier is just that people implementing these measures in different ways.
So you and I are going to talk more about this, but I think that because we actually just got funded to do a project to talk about this, but it's part of it is that it's not one thing. There's like what the people at Epic do, right? There are what people, who are clinicians do. But it's not, I mean, if you ask the clinician to reinvent the wheel, we will do it every time because we're used to working without complete data, right? Like that's, I mean, when we got Epic, someone handed us a bunch of we're talking about more complex data, but staging is very non-uniform in Epic. Diagnosis is fairly uniform. Staging, which we think is cancer, I mean, these are defined variables, values. In CNS, it's particularly simple compared to some other diseases. We're going to do it, and it's going to be a beast to even connect the difference between Seattle and CHOP's version of Epic. I don't actually think that's as much, and maybe that's because that's the hat I'm wearing, from the people at Seattle and CHOP. It's because I can tell you when we went live, someone sat in a room and said, would you like this staging form? And I said, sure. Like, I don't know what that is. And it was it was not actually created by someone who's a particular expert in cancer. And so I think there are things, but it's not that it's driven. If there was an available tool that was like, here's what you get, I mean, most of us will work within that. I don't.
Question or comment from the back?
Yeah, I'll say that I'm wearing a different hat than the NCI hat when I say this, just to be clear. So I was part of the RADx for underserved populations project. I was one of the 3 PIs that oversaw the data collection for that. We collected data on a lot of social determinants of health data consistently across more than 500,000 people, but it cost us a lot of money to do that consistently because you have to drive consistent questions across lots of different groups who historically don't work the same way. So I think there's a huge barrier to getting everybody to use the same questions. And Jeremy, your point about the Phoenix toolkit's exactly right. There's lots of great questions, but even around things like smoking history, there's probably 15 variants on how you ask smoking history. But I know that for those of you who do, well, you probably hopefully as pediatric oncologists, you don't have to ask about smoking history. But if you're an adult oncologist, there's really one way that everybody asks it now, but those older ways are all there in the toolkit. And so that's part of the issue. There's so many different ways to ask it. And when you ask them different ways, they're really not comparable. It's kind of like staging. I was going to earlier make a comment though. Now I'm wearing my NCI hat. We're with NCCR and with SEER, we actually do a lot of data linkage for everybody who is part of those registries. We pull in data, for instance, all residential history data from LexisNexis is available through SEER. If we can figure out how we can appropriately tie that to CBTN patients and fill in that residential history for their parents, we start to get a much better picture of some of those SDOH variables. And I guess my challenge to all of you is if we can do those kinds of things, how do we take advantage of that to understand the risk of those kinds of exposures where people live? Again, I don't know if it was this meeting or yesterday's meeting that I was in, but somebody talked about the importance of zip code DNA. So your zDNA, where you live really is important. So, again, getting that kind of information, and I think we can do that without having to ask the patients over and over again for the same data, right? So I guess that's I'm going to hold that out and maybe the carrot. And I'd love to work with all of you to figure out how do we do this and make it effective.
So it's a great point. I will say on the population science side, we sometimes use commercial databases like LexisNexis to try to track patients and get information on historical addresses before they enter treatment. And we have variable success with that. It tends to work relatively well for cancer patients because they generate a fair amount of paperwork, which is how essentially they're located. For etiologic studies where you also have unaffected individuals, you can introduce some bias because people who don't have health concerns that are as serious as cancer generate less paperwork and thus are harder to locate. But, I think the point about neighborhood broadly is a good one because it's probably simpler and I would imagine cheaper to get address data on patients than it is to get, sort of bespoke individual level data on social determinants of health. And there are increasingly a broad array of indices that are sort of purpose made to estimate, various components of people's social vulnerability or exposure to environmental contaminants, economic disadvantage that I think we could leverage, at least as a stopgap. Adam, do you have a?
I'm just going to put my Eric Durbin hat on. He's been part of some of our previous meetings. And, one of the things that surprised me a lot, and I think we've shared with the community here, is that, he and his team had identified that, this broad swath of Appalachia essentially had a much higher incidence than expected of brain tumors, as a context. And I think we still don't know why as a setting, and it's always this question, right? As part of the CBTN data release, actually, that's the first time they've been able to sequence the germline data of those patients, but it's the same question of how do we link across that data and the other social determinants. And I love that you brought this question into this panel because, I thought this panel was just going to really focus on germline, right? But in some ways, right, social determinants of health and germline are both risk frameworks, right, as a context that, should be added together, right, statistically in some ways. So.
Sorry, I was just going to say the word exposure is also terrifying. And I think of myself as a clinician 15 years ago when I used to tell people when they'd ask if they have to worry about their other kids, oh no, getting a childhood cancer is like being hit by lightning, I think was my blur. But now, we know that 1 in 5 kids has got at least them and maybe in their family, a known genomic cancer predisposition. So what do we all say now about exposures and whether cancer is preventable? Oh no, it's developmental. It's like being hit by lightning, and what I am now, I don't say that. I say we don't know because we don't know what we don't know. And so both social determinants and exposures are things we really haven't quite had the tools, but we're on the edge maybe of being able to look. And I didn't think in my career I'd be talking about cancer prevention for kids, but we might be having those conversations if we can get the right data to say, how preventable would cancer be.
I was just going to add too, even if we know that there's a germline predisposition, the accessibility of screening certainly has a huge impact on whether we're picking up cancer early. You think about things like Li-Fraumeni syndrome or CMMRD, where we know that screening significantly improves your survival benefit and your ability to catch cancer at a treatable stage. But if you can't actually get a whole body MRI because you live really far from a center or your insurance won't cover it, we're not going to pick up the cancer early. So I agree completely. I think we have to integrate the data for both germline and those social determinants and our understanding of what screening is accessible. And I think what the CMMRD community has also shown us is that partial screening is still some screening. And so being able to think about what is available and accessible to our patients will help us to treat them better.
But I don't think we should not look because we don't have the infrastructure to deal with it. And I think that, and I'm not saying that because of you, but I think it's really important. And this was a criticism about, wait, you're not ready for MCI. What do you mean you've got whole exome normal? Which, by the way, COG does not have any infrastructure to support your genetic counseling. We are a clinical trials organization. And that is not a concern that has been raised by anyone who has contacted us back, who's received their information. People were physicians wanting this information, whether they have all the resources they want to care for their patients or not. I think we would rather know, even if we're not quite sure how to do the best, than not know. I'm very much on that side of that argument, not that there aren't disagreements.
No, I agree completely. I just wanted to put in a plug for MCI and any tumor normal testing. We've doubled our ability to pick up germline predisposition by sequencing the tumor with the normal. So it's been a huge difference for our patients.
So we are coming up against the end of the time we have allotted, but I see there's one more question, I believe. See if we can get that quickly fielded.
No, please go if you're on the panel. I don't want to take your time. I can ask you all.
I was just going to say that on the phenotyping side, the social, the exposure, that's phenotyping. On the rare disease field, we came together, and after a lot of discussion, we end up adopting the HPO terms that other fields also use to be our standard. I think from what I'm hearing here, there is a need for the community to actually gather and have this discussion and decide what is the phenotyping that would be the minimal standard, how we would get that for pediatric cancer and make it available as a guideline.
Yes.
Maybe not everybody is going to be able to get everything, but everything is there on a guideline, on a best practice document of what to get and how to get, and people will get what they can.
Jeremy, Adam can get mad at me. Jeremy was an excellent moderator if we go over time. I just wanted to say that what you're saying really resonates in the concept of exposures and exposomes. So the other group that recently won ARPA-H funding, Gary Miller, Columbia University Medical Center, for the exposomes in farm work. And I would love to talk during the breaks with others who are working on this concept of a Venn diagram of all the different data sets, because Gary published something, and I'll have to send it to you, Adam. Actually, I tagged you in this about perfection can't be enemy of the good. Let's just get started. So we all want to scale and do things big, but what you said, it doesn't mean we shouldn't be looking, doesn't mean you can start, and you don't need all the data every time. But what is so hard is for each use case to create the Venn diagram of what your institution has, what the consortium has, what this one has, what the NIH, and so that Venn diagram, and then the bare minimum to get started, have some early wins. How do we get to that? And does it have to be domain specific, or can we be bigger? I don't know. I think exposomes getting added into the conversation with mass spec data is a great place to start, because that community should not be recreating the wheel of all the other data types that you've already all solved for.
Great point. Thank you. So I think we'll end it on that note. I found this very helpful. I think the panelists did a great job of enumerating some of the challenges we face, as well as some of the opportunities that exist for us to really make the biggest impact on patients' lives. So if you please join me in thanking the panelists.